described in 1959, and this is a liver phosphorylase deficiency. And that means that you're not able to convert glycogen to glucose monophosphate. There are two other glycogen storage diseases that they now put in the glycogen because they result in the same issue. One of them is what we used to know as type 8. The other is type 9. Type 8 was phosphorylase B kinase. The enzyme is phosphorylase, you make that. What? No, it's 8 and 9. There is no 10. There's no 10. There's no 10. There is a 0. We'll talk about that. So no, it's I'm sorry, it's 8 and 9. 8 is the phosphorylase B kinase. The numbers don't matter. What's interesting is what the deficiency is, the enzyme. The other one is protein kinase A, protein kinase A, phosphorylase kinase, one phosphorylase. So basically, you got generally the same issue. Interestingly, the liver phosphorylase is X-linked. It's not autosomal recessive, it's X-linked. The other two are autosomal recessive. And you're going to, you can imagine what the issues are. This is not quite as bad as the glucose phosphatase, which is type 1, but basically you're unable to, the patient is unable to take the glycogen stored in the liver and use it to maintain blood glucose. So what you tend to see with these patients is hepatomegaly, large liver. So they have, they're going to present with a large liver. Hepatomegaly. Low blood glucose. A real problem for physicians for a number of these diseases that are deal with liver glycogen is that the patient is going to have low blood glucose. They're going to be hypoglycemic. Low blood glucose to hypoglycemic, and not 2.5 and less millimolar is hypoglycemic. So they're going to be hypoglycemic, and they're going to have elevated ketones. And a lot of times, with some of these, not a lot of times, but with some of these, that hypoglycemia is associated with glucose in the urine. And so they start seeing a physician sees glucose in the urine and ketones, then they, no offense to physicians, but they tend to jump. And they're going to say, well, I got a diabetic on my hand. And you don't. You don't have a diabetic. In fact, that's really not the problem at all. That's also the problem with glucose tolerance test. You go into the doctor's office, you take after fasting, they give you they get a baseline on the glucose. They give you a known amount of glucose. They look at your rising blood glucose, and they keep you about 4 hours and check it over about 4 hours. How is what? They missed hours 6 and 8, and there's a sizable population out there that goes hypoglycemic after hour 4. So yeah, they look fine up to hour 4, but all of 7 hours, 6 or 8, they're hypoglycemic or severely hypoglycemic, so they don't do that well. We'll start today on this. You don't generally see liver failure. There's no real pathology in the liver. You just got a lot of glycogen in it, and it tends to swell. But liver failure does not appear to be a problem, like the other one where we tend to get cirrhosis, fibrosis, and cirrhosis of the liver. Treatment for all of these tends to be, well, high protein diet. Seems to work fairly well, and we're going to start on why that is today. And they like cornstarch, the uncooked cornstarch overnight. That seems to work well. This is also a good time to point out to you, well, type 6, yeah, but look at the different enzymes involved. And we're talking about infant onset, juvenile onset, late onset. Somebody said yesterday, well, if it's the gene, why is it different? Because the mutations in the genes are different. There's not a single mutation. Could be a variety of mutations that are going to show up at different stages in life. So there's been a lot of research now on what are the mutations. So they'll take patients and they'll do the gene screen and then do the sequencing of the gene and figure out where did that mutation occur, what's the amino acid mutation, or is there a piece missing, what's missing, and how does it present, how does that patient present compared to other patients that have quote the same type of glycogen storage disease. Kind of like being a diabetic. To me, there is no such thing as a diabetic. There is a disease, diabetes, and there's a big range on what you're going to see. And the same holds true here because you're impacting the gene a number of different ways. Type 7. So it must be running out of you only took care of 8 and 9 and then looked in. Type 7. This is a key. They have this one pretty well identified. And this one is phosphofructokinase deficiency. So phosphofructokinase deficiency, i.e., the individual can't take fructose 6-phosphate and convert it to fructose 1,6-bisphosphate. So there's an issue. So we have a hard time with glycolysis. There's several other diseases, genetic diseases, that fall in this kind of same category. We're going to present the same, and that would be glycerate kinase and glycerate isomerase. So glycerate kinase, if you don't have glycerate kinase, you don't take 1,3-bisphosphoglycerate, you get an ATP to make 3-phosphoglycerate, and if you don't have the glycerate isomerase, you don't take 3-glycerate to make 2-phosphoglycerate. So all of these are going to kind of come up with the same kind of symptoms. But this one's considered a glycogen storage disease because it impacts the ability of the liver or muscle to affect glycolysis. Generally, this is considered to be a muscle issue, and there are at least 4 variants of it that are known. It's an autosomal recessive trait, so it's autosomal recessive, and basically the person can't break down glycogen. The classical version of this is childhood onset. So classical symptoms are childhood. And basically what you see in the child is muscle pain upon exercise, and actually part of that pain is that during the exercise, since they're unable to use glucose, the muscle itself starts to break down. So the muscle breaks down, you end up with myoglobin in the blood, the myoglobin will be lost in the urine, and you're going to see a dark red to dark brown urine in these people, and that's the myoglobin showing up in the urine as you go through the kidney. So they're breaking down muscles to try and get energy. On occasion, some of these children show up with hemolytic anemia as well. So the red blood cell seems to be affected in some cases, which indicates that the PFK is absent in the red blood cell as well, or not very active in the red blood cell. It's possible that uric acid will damage the kidney because these people have a decrease in uric acid, and that can cause a couple things. Gout's one of them, but it can also cause some kidney damage as well. The way you diagnose it is muscle biopsy with phosphor or with PFK activity, and then of course there's a genetic study. They also look for creatine kinase in the blood. As that muscle breaks down, and that's what we're going to talk about when we get to muscle and protein, creatine kinase is released from the muscle. And creatine kinase is important in muscle because it provides you some short-term ATP, very short-term ATP, and we'll look at that when we deal with muscle. So that shows up in the chart as well. Then there's an infantile form, and infantile form is onset at birth. We'll notice it at birth. So in the infantile onset, you're going to know you're in trouble in a hurry. You've got a newborn that has myopathy, that is muscle weakness. It has flaccid muscles, hypotonia, not much muscle tone. These individuals are going to have a cardiomyopathy. They're going to get an enlarged heart because you're having the same problem in the heart muscle, the glycogen in the heart. And then, of course, PFK is required for heart functioning as well. So, a variety of issues, and there's really not much you can do, and the infant's going to die usually within a year of age. You're usually going to lose that one in about a year. They don't survive the first year. So, infantile is pretty self-limiting. The next one after that would be late onset. And in late onset, it is really not too bad. Late onset is pretty easy to deal with. There is some myopathy, and it's really the only significant feature. There tends to be some muscle degeneration. It's not a big problem in late onset, and late onset would be 25 or later. So that's not a big issue with that individual. The last one would be, there are some people that don't exhibit any of the muscle problems, and there's strictly a hemolytic anemia. So they have the third or fourth type, so a hemolytic anemia. All because the genes are affected differentially. Could be missing a part of the gene, could be an amino acid substitution. You substitute an amino acid over here, you're going to see early onset, you substitute it over here, it's going to be late onset. There's all kinds of things that can go wrong with that gene. Last but not least, type 0. And I think as we get better with genetic analysis, they're going to refine all of these glycogen forms. Type 0. Well, this is the one area we haven't been, and this is glycogen synthase deficiency. And it's pretty much limited to muscle. Glycogen synthase. And it's affecting the gene for glycogen synthase in muscle as opposed to the gene for glycogen synthase in the liver, and they're two different genes. So this individual has a difficult time synthesizing glycogen. It's inherited as an autosomal recessive. Upon fasting, these people become very hypoglycemic. You're going to usually see this early on in the child's life, usually about the time you start weaning or try to do overnight no feeding. So you start to do an overnight no feeding, start to break the cycle of feeding the infant every 4 or 6 hours, and you try to go overnight. What you notice in the morning is that you have an individual that's drowsy, doesn't seem to wake up well, could be pale, hypoglycemia leads to vomiting. So some of these people that have that are sensitive to glucose or carbohydrate because there's big elevation in insulin that drives their glucose down, and by hour 6, after the physician starts charting blood glucose, their blood glucose is in the toilet, and one of the things you'll see in these patients is that they're nauseous. They don't feel well. Drowsiness, pale, vomiting, fatigue, sometimes convulsions, sometimes fainting, true medical emergencies if you've got the glucose requirement for the brain. Also, sudden infant death is described as some of this, and it's because of the heart that the heart it's a heart issue, not a muscle issue, skeletal muscle issue, it's a heart muscle issue. So there is sudden death cardiac arrest that occurs as well. To make it worse, if you get through that period, the next thing that happens is that kids get sick, GI distress, some kind of GI bug, so they don't eat very much. They're going to become hypoglycemic in a hurry because they don't have any glycogen to fall back on. So these individuals will show up with some of the same symptoms. Early morning before breakfast, again, there's a fatigue, pale, vomiting, nauseated, possible convulsion, possible fainting, that sort of thing. And you find that they're low blood glucose. Over the long term, they may have slower growth, but not reciprocal. It's not a big issue generally. What else? This is one that looks like diabetes. If you exercise, there's going to be increased ketones in the urine, maybe even some glucose in the urine. The treatment for this is frequent meals. Small meals is what they use to help prevent hypoglycemia. You can sort it out with various types of testing. Sometimes it is liver, sometimes it's muscle. So they'll test both liver and muscle, do biopsy, do gene testing. Treatment, avoid fasting, keep your fast. Again, uncooked cornstarch and maybe a high protein diet. Because they all require glucose, one way or another. Which leads us, probably, to the next subject, which is going to be actually gluconeogenesis and discussion of how we make glucose. Since some animals don't absorb glucose, they're taking glucose to start to feed them to convert it to all the fatty acids. We need to sort out gluconeogenesis. Some of these patients are going to have to be highly gluconeogenic, and that's basically feeding them a high-protein diet. That also has an impact on meal frequency and timing for health and disease. And one of the, I don't know if you noticed, there's various faculty members that sort of wander through and listen to lectures and stuff. I think primarily because they want to see if they can figure out a way to get you fired. But nonetheless, one of them sent me an article from the Proceedings of the National Academy of Science, and I'm going to load that up along with the stuff you need to study to look at gluconeogenesis. And this is on meal frequency and timing in health and disease. It's an interesting article, and we'll talk about it on Friday. Probably even the quiz on Friday will be about that article. It talks about your meal frequency and how screwed up it is. That this is the way you were genetically designed to eat. So we'll look at that. So let's start with gluconeogenesis. And it's going to take you back to last quarter because you're going to have to draw it out. That's all these damn structures and enzymes that you get from glycolysis. So gluconeogenesis. All right. There is a requirement for glucose. There is a metabolic requirement. There is absolutely no dietary requirement. So it's a metabolic requirement. If we look at glucose in the body, we talked about glycogen. You're going to consume metabolically. The average consumption, how much glucose you need in metabolism a day, I don't know. I've seen crazy numbers, 120 grams, maybe up to 180 grams. And given liver, they figure that you might have something in the neighborhood of that available in glycogen stores. So The liver doesn't have that much glycogen, but they figure that that's just true. I don't think they have that much. 120 is going to be minimum. A lot of people will tell you that 120 grams is the glucose requirement daily for the brain, for nerve tissue. So that's the absolute requirement for the brain. And then the rest of it is for tissues that have an absolute requirement for glucose, and we're going to need to look at those. Body fluid only has between 10 and 20 grams of glucose, so there's not much there. So there's not much free glucose floating around. And the reason there isn't is because it's osmotically active. 10 to 20 grams, 20 I think is on the high side. So there's not much there. There are people that don't eat carbohydrates, and then there are ruminant animals that don't absorb carbohydrates. They're going to digest it and convert it to volatile fatty acids. We're going to figure out how they make glucose because they still have glucose requirements. Tissue requirements. Let's just look at those. Where do you require? Where do you have an absolute requirement? So the tissue requirement for glucose. Number one is going to be nerve tissue. The one on your list is nerve tissue, and that's the one that's affected the most with hypoglycemia. So you go to somebody and say you've got low blood sugar. And you can call as a first responder. Generally what you see with that person is they're agitated, irritated, they want absolutely nothing to do with you. Your job is to try and get glucose into them. And if you don't do that, you don't have to worry about it too much because they're going to pass out. And then you have a real medical emergency. So nerve tissue is going to be number one on the list. And as I say, they assume that somewhere in the neighborhood of 120 grams a day is required by the brain. Second one is adipose tissue. Adipose tissue has an absolute requirement for glucose. The liver doesn't. The liver can do what the adipose can't. And the requirement in the adipose tissue is that glucose is going to be converted to glucose 6-phosphate, which causes an ATP to a fructose 6-phosphate, and then it causes an ATP to a fructose 1,6-bisphosphate. With the aldolase, you're going to make glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And those are interconvertible with the triose phosphate isomerase. The dihydroxyacetone phosphate I'm going to draw that a different way because when we deal with that, I'm going to have the phosphate also on the bottom. I'm going to put the phosphate down here. Dihydroxyacetone phosphate, in the presence of glycerol phosphate dehydrogenase, and some NADH, and this is cytosolic. This is going to happen in the cytosol of the adipose, and there ain't a hell of a lot of cytosol in white adipose. The NAD is going to make alpha-glycerol phosphate, CH2OH, and we're going to get the OH and an H. So that's where your two protons, two electrons are going. They go down to number 2 carbon and H2. So this is alpha-glycerol phosphate or glycerol 3-phosphate. Take your pick. Alpha-glycerol phosphate, glycerol 3-phosphate is the precursor to a triglyceride. So what you're going to do